Relative versus absolute rates. M systematics P540. Differences in rates of divergence among lineages distract only from methods of analysis that require clock-like behavior of molecules, and alternative methods of analysis exist for all applications of molecular systematics except for the absolute estimation of time. Synonyms versus non-synonyms distance. Synonym sites are sites where a NT change does not cause an AA change. Only about 25% of sites so become saturated more quickly. Between points, more variations in non-synonymous rates within same protein, more variation in synonymous rates, which are used what is effect. Two cluster test. Estimate tree for each non-root interior node. Calculate average rate of both descent and clades. Test equality of rates. Usually variance or covariance of branch lengths doesn't appear to correct for multiple tests. Move up from leaves, eliminating a cluster if not equal. Finally, linear tree created. We estimate branch lengths under clock constant. Then neutral hypothesis. Most mutations have no influence on fitness of the organism. Advantages: mutations rare, deleterious mutations rapidly removed, greatest proportions of mutations have no effect on protein function. Rate of change is thus affected only by mutation rate and so should be relatively constant with the species. Variation in rate among genes between differences in selective constraints. Mutation rate in nuclear genes of mammals. Perfect molecular change, linear function time, rate constant, position, linear tree, perfect molecular distance estimated perfectly, calibration rates without error, regression time and substitutions error. Yang effect of evolution rate abstract. Yang calculates effect of evolutionary rate on accuracy of phylogenetic reconstruction. Stimulation study, branch length, expected total number and substitution per site not synonymous, estimates proportion of correct recovered branch partitions. Optimum levels of sequence divergence were even higher than previously suggested for saturation of substitutions, indicating that the problem of saturation may have been exaggerated. Then Bayesian parametric estimation. Density function for X given the training data set. For the definition of conditional probability densities, the first factor is independent of X since it must have assumed. Therefore, look at this equation. Then Bayesian parametric estimation. Instead of choosing specific value for theta, the Bayesian approach performs a weighted average over all values of theta. If the weighing factor is a posterior of theta, peaks very sharply about same value. Bayesian parametric estimation. The holy grail. Fossil evidence is sparse and imprecise or non-existent. Predict divergence times by comparing molecular data.